Good afternoon, everybody. May I ask you to all stand for the academic procession, please?
Good evening, everyone. My name is Loos and it is my great pleasure and honor to welcome you all to the inaugural lecture of Professor Ben Loos in my capacity as the Dean of the Faculty of Science. This week, the Faculty of Science has hosted a series of talks and events on the value of fundamental research in the natural sciences, in celebration of the International Year of Basic Sciences for Sustainable Development. And this evening we have another celebration, this time to celebrate the research and an important milestone in the academic life of Professor Ben Loos. So we are very honored that some of you are here in person and some online to celebrate with us. So I would like to extend a special word of welcome to Professor Deresh Ramjugarnath, our Deputy Vice-Chancellor Learning and Teaching, who is representing the senior management of the university this evening. I would also like to welcome our Vice Dean Research and Postgraduate students, Professor Frikkie Scholes and Professor Anna Mart Engelbrecht from the Department of Physiological Sciences. A warm welcome also to family and friends. Some of you are joining us online from Germany and Sweden. And I would also like to welcome colleagues from Stellenbosch University and other institutions. Many colleagues from the cell death and autophagy community are joining us online from other parts in South Africa, but also from the US and the UK. So a special word of welcome to all of you, and specifically Dr. Lucy Collinson from the Francis Crick Institute in London. Many of Ben's colleagues have become friends over the years, and you have also played an important role in his journey. Last but not least, I would also like to welcome current and former students of Professor Loos. It is now my honor to introduce Professor Ben Loos. So Ben was born in Berlin, Germany, and completed his undergraduate and postgraduate studies in physiological sciences at Stellenbosch University. He was appointed as a lecturer in 2011, and since then he has trained numerous undergraduate and postgraduate students. During his MSc, he began working extensively with fluorescence microscopy and led the microscopy unit of the Central Analytical Facilities, until 2010, when he handed it over with the super resolution confocal platform. Over the last 11 years, he has sought to gain a better understanding of the role of autophagy, that is, autophagic flux, in the onset of cell death associated with neurodegenerative diseases and gliomas. Ben's research focuses on Alzheimer's disease and neuronal aging, malignant brain tumors, as well as neuronal injury and trauma. To study the dynamics of the onset of cell death, he uses various sophisticated microscopy techniques. Aspects of Ben's work also leans into the field of data science, theoretical physics, engineering, and neural net approaches. And therefore, his collaborators include scientists from various disciplines. He You will hear much more about this research and also the spin-out company Phagoflux that was established in 2020 to develop autophagy sensing tools. Ben is not only passionate about research and teaching, but also about community engagement. In 2020, he was involved in a Science Meets Art project, where he and local artists worked together to address stigma and illness in a unique exhibition at the Rupert Museum. This exhibition is currently in the Jan Maton building, for those of you who have not seen it yet. Knowing that Ben is a physiologist, you may not be surprised to hear that one of his other hobbies is to build exploded skull models. For example, from Springbok or alike, which Ben describes as a smelly but satisfying anatomical affair. So Ben, we are looking forward to hear more about your work on autophagy and cell death control in neurodegeneration and gliomas. Please put your hands together for Professor Ben Loos.
Right. Good afternoon, colleagues, friends, family members at home. It's absolutely humbling to see you all here and sharing this work that we have been putting together through the last years with me. It's absolutely fantastic and very humbling. So thank you all for being here with me. I would like to welcome you by a welcome. Wamkile Kile. Herzlich willkommen, Familie zu Hause. There's a lot of family in Germany and Sweden. And so I would like to share with you today the role of a protein degradation process in cells and how it contributes to cell death decision making. And cell death decision making, of course, in the context of two major diseases that we are concerned with, which is neurodegenerative diseases, particularly Alzheimer's disease, and a very aggressive brain cancer, gliomas. So hence, the focus is on the role of autophagy and cell death control in these two pathologies. And I would like to dim the light because there are various fluorescent-based data that can just be appreciated a bit better. And I need to extend a wonderful thank you first to my many students that have actually done this very, very hard work molecularly to dissect. And so I'm very excited to share that with you now. I'm particularly thankful for my colleagues and friends that are here this evening sharing this time. So, now, not that long ago, we thought of the brain in this manner, where it was thought that language would be localized in this orbital region, probably because people with language would read a lot and spend many nights, or destructiveness would be just behind the ear. And now, of course, nowadays we have tools to unravel the molecular structures and cells that contribute to brain function, and particularly, of course, cells and circuits that contribute to various complex functions in the brain. And I have the privilege to look into living cells to understand which molecular processes control the decision-making of cells when they are exposed to stresses. And these stresses might be of a metabolic nature or other kinds of stresses. And Now, of course, we are reminded that the cell is the smallest living entity that is responding and that is dynamic. And once you have appreciated that under the microscope, you don't want to go back to anything that doesn't really capture that dynamic behavior of cells. I want to point out two structures here that we want to focus on, which are these magenta structures, mitochondria, very important for energy production, ATP production, and also the nexus for cell death decision, and then structures that are important for removal of toxic material. So I would like to stress that for us it's very important to think of the cell in the context of how long has the cell been spending in a particular adverse environment, and how would that influence its capacity to respond to a stress. And I would like to share here an image that we've acquired with Professor Craig Kinnear from the TB lab, many years of work together. You can see the cell is very sick. It contains a mycobacterium, but it is alive and it is responding to that stress. And likewise, the mycobacterium is using a range of molecules to try and hijack the machinery of the cell. So we need to think of the cell as a responding system and keep that very closely in mind. Now, this is important for the two diseases I would like to share with you. And this is on the left-hand side, a picture of what we find in Alzheimer's disease, where we have cells that die and also where we have aggregation of proteins, amyloid core structures that are typical for Alzheimer's disease. And on the right-hand side, we have glioma cells, so a brain cancer that is very resistant to chemotherapy, very challenging to treat. And here these cells are not responding and are not dying according to the treatment. Now, the molecular mechanisms that governs that cell death decision making is shared, and hence we can apply that for both of these systems. And so we have to be reminded that for the cell, energy is important and metabolites are critical. So the cell is continuously sensing the energetic charge, like the extent of battery power that is available in the cell, and it is continuously also sensing the abundance of metabolites, particularly amino acids, intracellularly. And when these metabolites decrease, then the cell will start to generate these double-membraned organelles termed autophagosomes. Auto, self, phago, eating, and soma, a small body. So small organelles that collect cargo, primarily long-lived proteins that need to be degraded, and that cargo is then delivered to other organelles, lysosomes, which we can think of acid bags, pH of 5, where that cargo is delivered to, fusion occurs, degradation happens, and the cell can then use these metabolites for energy production and synthesis of survival proteins. And this process happens all the time in the cell as it is sensing that process. Now, this is how this really looks like in a cell. So these cells have engaged heavily in autophagy. So you see here two cells, and you see how the cell has been generating a large number of autophagosomes, degrading substantial amounts of material. We can see that this can be really a major contributor to the cellular health. Now, this process, autophagy, is declining in aging. In neurodegenerative disease, it is dysfunctional, so hence the cell is not able to remove its toxic material. And in cancer cells, the cells hijack the system, and they have very high levels of this process autophagy to sustain their high metabolic demands. And so we spend time in dissecting and controlling that fundamental stress response pathway. I would like to show you here quickly when cells are exposed to stress. We see here cells that are where this signal disappears. These cells have committed suicide. Their mitochondria depolarize and they are not salvageable. So this is one point after which we are not able to save these cells. And we spend time to understand why are these cells here dying earlier and other cells later in this context. Now, how can we understand these various components that are important? So this is here a team in the Gordons Bay Triathlon ready for the swimming. And one can appreciate if you are well trained, you may be well prepared for this race. But if the weather is bad, the water is icy, the water is choppy, it might be challenging to complete that in an appropriate manner. And likewise, if you're ill-trained, you will suffer. And the same principle is important to understand the cellular stress response. So we have to very carefully understand what is metabolically happening in the cells, what kind of activities are going on, what metabolites are preferred, and also what is the magnitude and duration of the stress response of the external environment. And that will contribute to that decision making. So hence you can see it's complex and many factors need to be considered. So I would like to first, before describing the two particular processes in Alzheimer's disease and gliomas, share with you this protein degradation process, autophagy. So these green structures here are autophagosomes that collect and remove cargo. Now, it could now be that this cell has been very active and has generated a lot of these autophagosomes to remove toxic material, which would be a favorable thing. But it could also be that the cell is not able to degrade this cargo and to receive these autophagosomes due to a dysfunction, for example, in the lysosomes. And hence it would be a pathological scenario. So these are, of course, two completely opposing systems. And how can we understand the activity of that process and what is really going on in the system? And here, through many years of wonderful collaboration with Prof. Janni Hofmeier and Dr. André Dutour, we have indicated a system where we actually measure the activity as a rate of degradation, which we can compare a bit like water flow in a basin. And you open the tap. And when you just take a picture of that running tap, you don't really know the rate of that water flow. But if you put the plug into the basin and you record how rapidly the water rises, that you can express as an activity in milliliters per minute and so forth. And this is exactly what we have applied to the single cells. This is a single cell that is showing here the movement of the autophagosomes. And what we then do is we inhibit the fusion between autophagosomes and lysosomes. In other words, we put that stopper into the basin, and we measure the rate of accumulation, and can therefore indicate how many autophagosomes per cell per hour are being generated or degraded, as a very precise measure of autophagy activity. Now, this was a tricky concept in the field. To understand that the number of these autophagosomes in a cell does not determine the activity. So you can have cells with a low number of autophagosomes, but a very high activity, or cells with a lot of autophagosomes, but a low activity. And with this system, we can really very carefully quantify this process, and it's now on the single cell level a very finely describing method. Now, why is that important? Because these autophagosomes degrade material. They degrade long-lived proteins. 99% of the cellular material are long-lived proteins. But more importantly, the autophagosomes degrade toxic proteins. And in Alzheimer's disease, the toxic proteins are amyloid beta, phosphorylated tau, so particular proteins that lead to cellular stress. And when we use advanced imaging techniques and we stain for proteinaceous cargo, these red dots, then you actually see that there's a lot of cargo present, but not all of that cargo is actually targeted and removed by the autophagosome. It's a little bit like in the COVID time, we had lots of aeroplanes that were flying empty. And here, just measuring the autophagy activity is very important, but we need to align it with the cargo that is disease-specific. And we use various techniques here, including theoretical physics with Professor Müller-Nederbock, to understand how energetically favorable are these processes and how much does it cost the cell to do that. Now, as we age, the autophagy activity declines. And in fact, aging is the biggest risk factor for the onset of neurodegenerative diseases. And that's, of course, a major challenge for us. A, we are aging, we are living longer. And B, the lifestyle-associated challenges, like diabetes and so forth, contribute to poor aging. So hence, we have a major dilemma with very high numbers of neurodegenerative diseases. And now, how can we now move this concept into the Alzheimer's disease field? So I'd like to share with you how we approach that scientifically. In Alzheimer's disease, we have neurons that start to die due to toxic protein buildup. And when sufficient neurons have died, the first symptoms of mild cognitive impairment set in. This process, however, starts very early on. So you and I could already have major dysfunction in the system and may have the first symptoms very soon, and we are not able to measure it. So how can we dissect this in the lab? We are using here cells where we can switch on the key proteins that drive Alzheimer's disease. In this case, amyloid precursor protein. You can see here over time, the cell becomes green here, so it expresses a lot of this particular protein, which toxifies the mitochondria and various components in the cell. Now, importantly, as soon as that happens, the cell is engaging with the autophagy machinery to remove the toxic material. And we have found that the cell is functional in this process, autophagy, all the way until it is starting to die. So in other words, here, when we look for markers for cell death, we can see that there are markers for apoptosis that are activated whilst the cell is still able to degrade its material. So that means if we now look at the cell at a late point in time, this would be something like severe Alzheimer's disease, we suddenly see here signs for autophagy and apoptosis and cell death, and you are not sure why is this dual picture here. Of course, it is because the cell has responded to it. And the good news is that it means that at any point in time when we target this process, we are able to still reach the cell therapeutically. And hence, it is good news. So the cell is kind of dying whilst fighting against that proteotoxic load. Now, of course, we have to bring this always into an in vivo model system. And here we use rodent models. We create here a condition of major neurodegeneration using paraquat. So this is hippocampus tissue, and the cells are dying here heavily. And now when we expose these rodents to enhancing autophagy by allowing them to be calorically restricted. So in other words, we take away their food. So we allow them to fast for 2 days. Then they are allowed to feed ad libitum again. Then they are fasting. Then they are feeding again. And we see, and in this process, we enhancing substantially autophagy activity. And we see that in doing so, the cells are substantially protected, purely by enhancing this autophagy process. And this model system is a very crude system. So taken together, we can enhance autophagy by decreasing metabolites, so through fasting or skipping a meal or having delayed breakfast. These are all autophagy enhancing interventions. And we see substantial translation in the neurons when doing so. Now, I would like to briefly switch gears and indicate here we have a problem when it comes to imaging, and that is resolving power. So when you land in a city, then you may see that those areas where there is substantial amounts of light, you are actually not able to resolve the houses, right? But if you would land in Cape Town at night, at the moment, hopefully not tonight, but kind of in our current context, there could be a different scenario. If there's load shedding, you would actually see that regions will be switched off. And if you recorded that over time and you map that, you would resolve the structures so much better because we are allowing the lights to basically come on and off in that context. And this is what we do with the cells. So we are overcoming here the limit of resolution by letting the molecules blink. So every single molecule emits here a signal, and that means when there are two molecules that are too close to one another, it doesn't matter because one has been switched off, and in the next image frame, the next one comes on, and that allows us to break the limit of resolution. And now, when we do that, we are able to enhance our resolving power 10 times. So it's a substantial improvement. We see a very fine tubulin network. Why are we doing that? Because we are interested in the proteinaceous cargo that the cell needs to degrade, and we need to measure it finely. You see that everything that we are dealing with all happens way before the cell actually dies. And hence we need these very sensitive tools to be able to see how sick the cell is. This is one method. We see this is a cell with single proteinaceous clusters here. And when we inhibit the process of autophagy, you see how immediately these clusters start to increase, only 2 hours after inhibiting this process. So we can characterize this very finely with regards to the molecular density that is in fact presented here. And when we do that for the autophagosomes in green and the cargo, you see here this material under normal conditions, right? All these red spots is material that the cell needs to remove. When we inhibit the process of autophagy, that material very quickly builds up. and aggregates. So it is like the refuse is not removed from the household. And of course, if that happens for a long time and the municipality is maybe on strike, then that leads immediately to major disruption and later, of course, disease and so forth. The same system is applicable in the cells. Now, in the Alzheimer's disease case, we see that when we switch on Alzheimer's disease in the cell, these are all toxic proteinaceous clusters, APP protein clusters, in the super-resolution mode. And when we use an autophagy enhancing therapeutic drug called spermidine, which is an amine found in peas, for example, or cheese, has received a lot of attention recently, we can bring these clusters significantly down. So we assist the cell really to remove its toxic proteinaceous clusters. We can quantify that and understand that the cell, through this mechanism, is saved and is not degenerating and dying by enhancing autophagy and by assisting the cell to remove the toxic material. And in vivo, if we in a rodent model, again here you can see tissue from hippocampus from animals that are highly diseased. You can see the proteinaceous aggregations. When we provide the spermidine in the drinking water for 3 weeks, we have a major reduction in the stress markers. So very powerful. The inherent autophagy machinery is very powerful to really impact cellular health. Right. Now, there is another problem I would like to share with you. And that is that we often use electron microscopy in the field because it has fantastic resolving power. And we see the nucleus and various structures. And you see this would be here a cell in the context of Alzheimer's disease. You see all these structures here accumulating. But you don't know what they are. You may recognize mitochondria and maybe Golgi, so particular organelles in the cell, but you don't know their molecular identity. Now, in the fluorescence, we can do that. We can label specific structures and unravel particular molecular identity. So how would it be if we could bring these two modalities together? And so what we would do is we would seed our cells, and then we would select cells that we would like to acquire and image. But we use very specialized dishes here that if you look carefully and if you kind of lean from the side, you see that there's actually a grid pattern. And you see there is here Q2 and Q1, and you have to kind of lean from the sides and Q1, P2. And if you do that and then someone walks by, they think you have, now something is strange here. We do that to see the exact location of that cell so that we can then zoom in so we know, yeah, that's the cell of interest. That's what we acquire, and we acquire what we wish to see. Here, for example, you see one particular organelle. Here you see an organelle where an autophagosome has already fused with the lysosome. Hence, there is some blue coloration here because the pH there is lower. But we don't know how that cargo looks like. What kind of cargo is in there? So what we then do is we take the sample and we embed it in plastic and resin, and we break it apart, and we take a saw to trim into exactly that cube where that cell was positioned. And then we take it to another microscope. We drive there with a car, and then very carefully section through that sample in nanometer step widths, because it's only a single layer of cells that you have there. So if you would normal techniques, you would section that cell off and it's gone. So very, very carefully, and then you generate signal, and lo and behold, you are able to find exact the same structure, and you can bring these two together and therefore bring the molecular identity to this high resolved electron micrograph. And so we can see here now and appreciate the extent of this cargo that we see here. Now again, why is this important? It is important because we need to know what that cargo is. In Alzheimer's disease, how much amyloid beta, how much phosphorylated tau. In Huntington's disease, how much Huntington. In Parkinson's disease, how much alpha-synuclein. So various cargo, and we need to be able to align these two. So what I've shown you here in 3 minutes took about 3 to 4 years. Dr. Creel has done wonderful work to set that up because it's a very technical process, and we are now able to characterize our model systems in that precise manner. We can do this three-dimensionally to describe that cargo that we see, and we can screen the various molecular identities we are interested in that particular highly resolved context. And so this was a wonderful collaboration through Lucy Collinson from the Criggs Institute for us to be able to get through this incredibly technical, challenging process. And I see now here this is not presenting properly, but that is fine. So when we do this in the 3D context, we are now able to say to what extent is an autophagy enhancement clearing toxic cargo on a highly precise manner, so that we can say what type of drug should be prescribed, what concentration of drug should be prescribed, and is that drug in fact removing the cargo associated with the disease. Right, now I would like to briefly switch gear and speak about gliomas. So in gliomas we have the challenge that these cells are resistant to chemotherapy. The treatment is very poor. It's a very challenging disease. The cells of the glia in the brain infiltrate, and hence it is very challenging to resect this tissue. And now these cells have very high autophagy activity, and there is globally a lot of interest to exploit the system to sensitize these cells to undergo cell death better. And here we grow, this is here a human brain cancer, and these colors here indicate low and high autophagy activity. And in using this model, we're able to say which of these cells die first and are most sensitive to a given treatment. So it is very close to the patient samples. And of course, that is what we always wish to recreate in vitro. And now, one of the very important organelles in the cell are the mitochondria. And these mitochondria generate ATP. They are the powerhouse of the cell, but they are highly dynamic structures. And in order to understand whether a treatment can kind of trip these cells over to death, we need to measure the mitochondrial function. And we do this here by, if you look carefully, photoactivating, so we make visible just a small, this is a cell, and can you see this is very bright and this is very bright. These are just a few mitochondria in the cell. The cell is distributing this protein through the rest of the mitochondrial network. So this is how dynamic this mitochondrial network is. And we can now use this tool to measure the susceptibility of the cell, how easy they die and how easy they undergo metabolic failure. And we have found that if we again tune autophagy activity, if we enhance it, we cause metabolic failure because it is already very high in cancer cells. Then we inhibit it and present the chemotherapeutic drug. Then these cells are dying most significantly. We would not be able to dissect that with some of these techniques. So taken together, both of these pathologies with the metrics of autophagy activity and mitochondrial quality control, so the energetic part and the protein degradation part, there is hope to target the molecular defect much more precisely. So in going forward, one of the big challenges is to translate this closer to the clinic. So we're working very hard on being able to measure this process in vivo, which is currently very hard to do, so that we can see what is the autophagy level in this particular cancer in the patient. And so to briefly summarize, of course, it's incredibly difficult to choose what to share here. I just want to very, very briefly indicate that there are many other areas of concern that we are working with, such as neuronal trauma, metal neurotoxicity related to our mining environments, manganese and cadmium and so forth in South Africa. Then how to use fluorescence to assist in the surgery. So we work with the neurosurgeons to assist how we can make better visual the tumor that is being operated. And in the Alzheimer's disease field, how we can pair the current drugs for better treatment outcome. So being aware that I have only 4 minutes left, I would like to now, in the last few minutes, share with you very brief examples of the joy of science when we step out just a little bit out of our fields of focus. And so the first one, with wonderful work with the engineering department, Prof. Harold and the team, we have started to develop sensing technology to measure this process, autophagy, rapidly and sensitively. Now, this device was was a starting point, and we have now fine-tuned that with the aim that you and I one day can measure the autophagy activity, how well we are aging. When we exercise, autophagy is enhanced, that we can track and measure that not just in the research and pharma field, but also in the wellness field. A second problem I want to briefly check in is you have seen mitochondria and the dynamics of these mitochondria. And so you can see cells can have fragmented mitochondria and cells can have more fused mitochondria. And they do that to align again themselves with their requirements for energy. And that's a very rapid process. It's currently there had been no way to measure that process. And now in several diseases, in Parkinson's disease, for example, we have major changes in this network, and we have no tools to quantitatively measure that. So here, with a team of Prof. Thomas Niesler and Dr. Tjart, who is also here, we have developed software technology to, in the living cell, pinpoint the regions where two mitochondria merge, fuse, and where they are undergoing fission, where they are separating. And we can now really, in 3D, in the living system, calculate that and understand whether the cell is in equilibrium with these two processes, or whether it is, in fact, moving towards fusion or fission. So a very, very sensitive technology that we are now applying to our systems. And then you see that when we appreciate the living cell, it is very difficult to actually go back to a picture like that, where you see the mitochondria and you see structures there. And we thought we need to actually bring this into the teaching environment and to give our students a head start of understanding the disease cell and those pathologies that develop in the cell. And so here we have what could be not better with using technology that the youth, the students, are very often using in any case, which is virtual reality-based tools. And here, Dr. Tjart's team and Heino Nopio, who is also here, we have developed a tool where we can now, with real data, step into the cell, kind of pull the organelles, the various organelles of interest to you, click a button where you see the dynamics and you have the information that's suitable for the undergraduate and postgraduate so that you really understand the dynamics and the cell as a living system. Very, very exciting and lots of fun. And then lastly, I would just end off with that what we are very poor with is appropriate science communication. And of course, the diseases that we are working with are challenging diseases, and there's of course a lot of pain associated with it. And the students over the years always indicated we need to better share what we are doing with the communities at home. And we are, of course, aware of, specifically in neurodegenerative diseases, the issues of stigma. When there's forgetfulness, then it might be madness. And then there might be exclusion of that person, and very often women, in that community. So here we have created a science material and paired up with local artists, with Prof. Elmarie Constantios here. And the artists were inspired by the topics, created particular artwork, which we then translated as a first point of departure to better share those pathologies that we can visualize and let the picture just speak so much better. And that, colleagues, I'm 2 minutes over. So I would like to thank my students, my stars through the ages that have done this fantastic work through the years, many, many, many hours on the microscope to an extent that one can develop the ocular structures on the glasses. So it is absolutely appreciated, and I would like to thank the Faculty of Science, the leadership of the science faculty for the many years of support. Many times I've been in your offices for various questions, grants, and concerns, and have received wonderful support, so thank you very much. My many mentors and teachers, many of you are here, many are online. It's an absolute privilege to have walked this path and walking this with you. So thank you so much. And my funders, of course, and very importantly, my dear family, Unati, Joshua and Aaron at home. They all know about autophagy. They are not allowed to have Nutella in the week, only on Sunday, because high glucose levels blunts autophagy. So, my dear friends and colleagues and family at home, my parents, liebe Eltern, herzlichen Dank. Thank you so much. It's much appreciated.
Thank you very much, Professor Loos. That was an absolutely fascinating lecture. Good evening to the Loos family and friends, leadership of the Faculty of Science, colleagues, ladies and gentlemen. It's an absolute privilege and pleasure to be with you tonight, to be part of and share in a rite of passage in academia. The recognition of a colleague entering the ranks of full professorship, and them sharing through their inaugural lecture their innovative ideas, their discoveries, their deep insights but importantly, doing it in a manner where it's not just sharing it with the academic community, but also with the public. Now, thank you, Professor Loos, for the fascinating journey into the functioning of the neurocell, and specifically about the cellular fate. The ability to understand cell fate and potentially control it has profound implications in the management and treatment of a number of the most cruel and debilitating diseases, such as Alzheimer's and gliomas. Now, one of the indicators of a really good inaugural lecture is the ability of the professor to share deep and complex disciplinary knowledge in a manner that is both understandable to the general audience, but also clearly articulates the novelty and the complexity of the work to disciplinary experts. Now, this you have done incredibly well. Getting that balance right is really challenging, and you have done this tonight. So, phenomenal inaugural lecture. In your lecture tonight, you introduced us to the concept of autophagy, the most fundamental systems of cells to respond to stress and metabolic perturbations. You then introduced the concepts and definitions of measuring autophagy activity, as well as linked decreased autophagy with aging and the increased risk for the onset of neurodegeneration. Thereafter, you shared your work and thoughts on autophagy control in neurodegeneration, as well as autophagy control in gliomas. Now, your lecture gives us hope that with improved assessment tools, techniques, and approaches, we may be able to better understand neurodegeneration, and hopefully in the future, near future, through that find treatments for Alzheimer's and for gliomas. Now, it took me back to my days of studying biology in high school. And I have to say, if I had a teacher in high school that was able to explain the concepts the way you did it tonight, I would have readily followed a career in physiological sciences as opposed to engineering. So congratulations, Professor Loos, on your incredible body of research and work in the field of physiological sciences, and in particular in autophagy. Thank you for sharing your insights on this fascinating topic. Your research is bringing incredible insights to autophagy through some of the things that you're doing, through the sensing devices that you are developing. I'm glad to see that I'm one of the proponents for innovation and entrepreneurship, that you are translating your research into products and tools that will be commercialized, not just because you want commercial value, but to advance the field of understanding autophagy and ultimately to have improved human wellness. Congratulations on your work once again, and we look forward to the cellular impact that your research will make. Well done.
Ben, it is a great pleasure and privilege for me to congratulate you on behalf of our department and the colleagues in our department for achieving this great milestone in your academic career, and also to thank you for an excellent presentation. And I think you all will agree with me that it's clear from the presentation that Ben is an outstanding scientist, but he's also a passionate teacher, but would love to make meaningful connections with his students where he uses science to have an impact on their lives. He also plays a vital role in our department as our honors coordinator, and for the past, I think
5 to 7 years, you were already honours coordinator, and our honours student amount of honours students grew from 5 to 7 per year now to 20, plus minus 20 students per year under Ben's leadership. And to put this huge contribution of Ben into the field of microscopy into perspective, I need to go back a little bit into the history. And I think it was 2004 when Ben was still an undergraduate student in our department. And after histology lecture, he came to me and he said to me, "Yeah, he just loved histology." And no students have ever told me that. So I thought, yeah, this must be a very special student, because students don't like histology because they cannot see the pictures very clearly, so they cannot distinguish so greatly between the different types of cells and the different types of tissues. So and that was the beginning of our journey with histology and microscopy together. And yeah, so in 2006, our department acquired an Olympus Live Cell Wide Field Imaging Microscope, and Ben was then an MSc student at that time and very passionate about microscopy, and then he was appointed to manage the system. And a year later, the cell imaging unit was established as part of the Central Analytical Facility, which was then also managed by Ben. And at that time, he was also working on his PhD while he had a full-time job in the microscopy unit. And these were very exciting times when the first data sets for techniques such as fluorescent resonance energy transfer, or FRET, were published, and it became clear that the research environment needed more and better imaging equipment. So Ben also managed to develop very successful collaborations with a wide range of colleagues all over the faculties, and a lot of you are sitting also here in this evening. And it's through one of these collaborations in 2010, with an increasing demand of the growing imaging community, the request for a confocal microscope by Ben and Prof. Bert Klumperman of the chemistry department was granted, and the confocal microscope with super resolution capacity was then obtained. And the successful acquisition of this Zeiss confocal microscope in 2011 changed the landscape of fluorescent microscopy forever in the university and in the department. So as colleague, Ben is really a much loved and respected by everyone, and with his kind and charming and caring nature, his strong values, honesty and empathy are some of his most valuable attributes. So he's not also only making a difference at university level, but also in society. So as a student, and I think it was still he was a master's student, he and his wife and then another student friend of them started in a Kayamandi organization, Vision K, which has expanded later to become Vision Africa, where they focus on leadership development, academic improvement, and active community participation for the youth. So I will conclude with two quotes very applicable to Ben. So the first one is from Maya Angelou, and she said, "I've learned that people will forget what you said and people will forget what you did, but people will never forget how you made them feel." And Ben, you are one of those handful of people that listen with all your attention when you speak to people, listen when people speak to you. And I very often also see it with your postgraduate students sitting in your office, and that you really listen with attention, and this makes them feel heard and also makes them feel that they matter to you. So thank you for that. And I think it's very appropriate that I end with a quote from a German scientist. So most people, and he said that most people say intellect, that's intellect that makes a great scientist. But Albert Einstein said, no, they are wrong, it is character. So I just want to thank you for being such a great role model and for making a difference in the lives of so many people. And we are looking forward with great expectations to your future contributions to the department and to the faculty and also to the university. So finally, on behalf of Ben, I also want to thank the Stellenbosch University team, especially Ms. Amira Brown, Jonathan Dale Blankenberg, and Letitia, who assisted with the lecturing, editing, and printing. And in Ben's own words, they were awesome. So thank you all for coming, and you are most welcome now to come and join us outside for something to eat and to drink. So thank you very much.